Good day, everyone. Today, we will be speaking about protein sequencing. So, we will take a look at what the proteins are, how the proteins are made up of, and why we need to know the sequence of proteins or how protein sequencing can help us achieve various targets. So what you're seeing right here on the screen is a simple polypeptide chain which is comprised of many amino acids which join together to form a polypeptide chain. And as you must be aware of, amino acids are the building blocks of proteins, and amino acids are linked together by peptide bonds to form the polypeptide chain. And we know from the protein structure that the proteins can have four different structures. Primary structure, which you are seeing right over here as a polypeptide chain, then they can undergo many foldings to form the secondary structure, the alpha and the beta plated sheets. Then they have the tertiary structure when they get their three-dimensional structures, and multiple types of polypeptide chains join together to give you the quaternary structure. So how exactly these structures are being formed, we have already discussed. Now today we will be discussing about how these amino acids and the way in which they are arranged, or the particular sequence in which the amino acids are arranged, give the protein a particular sequence, and how we can understand in which sequence these amino acids are arranged in a particular protein. So as I said, proteins are composed of linear chains of amino acids. They are linked together by peptide bonds with the amide bonds. So how are these peptide bonds formed? They are formed by linking together the amino functional group, which is present at one end of the amino acid, with the carboxylic acid functional group at the other end of the adjacent amino acid. So one free amino group of one amino acid will react with the carboxylic acid functional group of the adjacent amino acid to form the peptide linkages. And likewise, they arrange themselves to form the polypeptide chain. So that means a polypeptide chain will always have one free amino group at one end, and the other end will have a free carboxylic acid group. The end which has a free amino group is termed as the N terminus or the amino terminus, and the one which is carrying the free carboxylic acid group, we term it as the carboxyl end or the C terminus. And these two terminus of the polypeptide chain is important as far as the protein sequencing methods are running around these two terminus. Either you can read the sequence of the protein from the N terminus to the C terminus, or in some methods of protein sequencing, we start reading it from the C terminus to the N terminus. But more frequently, we start our reading of the protein sequence, the amino acid sequence of the proteins from the N terminus to the C terminus. So let's check out how it is being done. Proteins, we just saw, they are biomolecules, polymers of amino acids, and the structure variation also affects the function of the protein. We have seen this. So that means if there is a difference in the amino acid sequence in the peptide chain, that could lead to variation in the protein structure, which could ultimately result in a changed function of the protein. So that means the way in which the amino acids are arranged in a polypeptide chain to give the protein a particular sequence is very much important and very much related to how the protein is coming up into its final structure and how that structure is helping in the function of a particular protein. So, speaking about the applications of protein sequencing, or what exactly is protein sequencing? It is a method by which we can find out the sequence of amino acids which are present in a particular protein. And once we understand the sequence in which these amino acids are present in the protein, that could help us in understanding or predicting how this protein could achieve a final structure. Once we know the structure, we can elucidate the function of that particular protein or the location of that particular protein in the organism, and it can also help us find out the location of genes which are coding for that particular protein. Then in functional genomics, or it can help us in identifying the protein folding patterns. And we know that many of the viral toxins that are injected into the body by viral infections, they are mostly in the proteinaceous form. So making drugs which could combat all these microorganisms or these microbial toxins, so understanding the structure of these proteins and the sequences of the proteins could help us to elucidate more effective drugs, as well as in antibiotic production, in recombinant protein synthesis. So the applications of protein sequencing is so very vast. So let's check out how exactly we can find the sequence of the proteins. So there are many different methods for sequencing proteins. The most common or most famous being the N-terminal sequencing, otherwise known as the Edman degradation method, formulated by for Edman. And C-terminal sequencing is there, or you can either know the DNA sequence, and from that we can deduce the sequence of the protein. Prediction from the DNA sequence is also possible, and nowadays we can have automated protein sequencing is also possible. And the whole story began by the first protein sequencing of insulin by Frederick Sanger in 1953, when he tried to elucidate the amino acid sequence present in bovine insulin. And for his work, he was awarded Nobel Prize in 1958 in chemistry. So protein sequencing is used to identify the amino acid sequence as well as its conformation and how it is affecting the structure of that particular protein. So once identification of the structure is done, we could also deduce the function of the protein, which is very much important to understand the cellular processes that is taking place within a living organism. So this is the all cheerful Sanger, who is celebrating his first Nobel Prize, and he's best known for winning twice Nobel Prize in chemistry. One is for his deducing the amino acid sequences of insulin, and the second, again, he has played great role in DNA sequencing as well. So this picture that you are seeing right over here is one of his laboratory notebooks when he was working with amino acid sequencing, that is insulin, and many of the nodes and the things that he has noted, you can see here dotted lines, which could have been the positions for the disulfide linkages in this protein molecule. So it is his work that has laid the foundations for what we know today as the protein sequencing. And till today, even if it is the day of automation, the things that has come out from Sanger, taken up by Edman and the followers, has laid the foundation for protein sequencing as we know today. So this is just Sanger's degradation procedure. We will not be speaking much about Sanger's degradation process. We will be focusing more on the Edman degradation process for today's lecture. But just to give you an overview, Sanger's degradation procedure for sequencing insulin was in such a way that various samples of the protein were broken down into fragments of bits by treating them with acids. And these small peptides fragments were then looked out for understanding the amino acids, and with different fragments, the overlapping fragments were found out. They were again broken down, chopped down into amino acids, and paper chromatography was used to separate all these things. And finally, the whole picture was made clear by putting together these overlapping fragments one over the other, and finally making the readings. But the problem with Sanger's degradation procedure is that at every step you are degrading the protein as a whole at every step. So acid hydrolysis was the main process that was being done. So you needed lots of protein samples to complete the sequences, and that in itself was a great task. So having a big amount of protein samples was a basic need, and degradation that is taking place, or you are losing your protein samples across this procedure. So people started thinking, how can we better use the protein samples that we are having to understand the protein sequences or the amino acid sequences in the protein. And that is where the Edman degradation process comes into play. And the N-terminal sequencing method that Edman formulated, or analysis of the N-terminal sequence using a reagent known as the Edman reagent, has come into play. And along with the newer technologies like chromatography and mass spectrometry, protein analysis and sequencing has become more sophisticated and more reliable. So well-established sequencing method. This provides accurate protein sequence data, and sometimes it is compared to the mass spectrometry, it gives even more powerful or accurate protein sequence data. So this is for Edman, who is the person behind this Edman sequencing method, and we call this process as Edman degradation process of amino acid sequencing or the protein sequencing. So the general gist of this whole process is that the method uses a label which will go and bind with the N-terminal amino acid to form an amino acid derivative, which could be cleaved out of the polypeptide, leaving behind a shorter intact peptide chain. And using these amino acid residues, which are now linked with the Edman reagent, that could be fed into chromatographic techniques to identify the amino acids. And again, the cycle could become repeated till the last amino acid of the peptide chain is understood and what it is could be deciphered. So Edman degradation reaction is nowadays automated. It got automated in 1967 by Edman and his colleague Bex. And nowadays, the automated Edman degradation, or the protein sequenator, that's what we call it today, the protein sequenator, is widely used. So this method can be used on peptide chains which are up to 40 to 50 amino acids. Beyond that, if it is really big protein that we are trying to work with or trying to understand the sequence of it, then we need to chop it down into smaller fragments and then go back to the Edman sequencing program. So let's check out the details of how this Edman degradation process is being done. So this is a schematic workflow that is used in a sequencer or as a whole for understanding the protein sequence. So everything starts with a sample. That means you need to have a purified protein sample. And once the sample is purified, if it is a really, really big protein, then you need to go for denaturation or digestion with appropriate methods. It could either be high temperature treatment, acid treatment, or even certain enzymes which can top down proteins at regular positions or at specific points. As we do the chopping of the DNA with restriction endonucleases, we do have enzymes which cut the proteins at specific points. So once the purification, denaturation, and digestion of the protein is completed, then comes the labeling of the protein. So here we are using N-terminal labeling, where we are using Edman reagent, which we call, which is chemically phenyl isothiocyanate. So using this phenyl isothiocyanate, we label the N terminus of the peptide chain we are trying to sequence. So this phenyl isothiocyanate will go bind with the amino part of the N-terminus amino acid, and it forms a conjugate. Okay, so once this is done, This is usually done under a slightly alkaline condition. And once this is done, then uh, by changing the pH of the medium and by adding certain other reagents, we can cleave this polypeptide chain, thereby releasing this phenyl isothiocyanate along with the first amino acid. So now you will have your second amino acid at the N terminus end. So that will now be the free N terminus. So what will happen to the first one that has been removed from it? That could be fed into chromatographic techniques for identification of the amino acid. Either you can use chromatographic techniques, or you can couple it with mass spectrometry, where mass by charge ratio will help us identify the amino acid by comparing it with a standard graph or a standard spectrum of already known amino acids. And finally, this what happens to the rest of the peptide chain that has been shortened now after the removal of the first amino acid? Now you have the second amino acid at the N terminus end. So now what can we do? Now the reaction proceeds in such a way that again the Edman reagent will come, label the second amino acid, you can cleave it. Now again you have your shortened polypeptide chain. Then again this continues till the last amino acid has been fed into the chromatography system, and you can go for your data analysis. So this is in a nutshell the schematics of how the workflow is done in the sequencer, where we are using N-terminal labeling using the Edman reagent. So going into the details of all this, so once you have your protein sample isolated from the sample, you can purify it using electrophoresis, either one-dimensional SDS page, that is sodium rhodesyl sulfate polyacrylamide gel, or even you can use the two-dimensional gel electrophoresis for separating out the protein of interest. And once this is done, you can either use the protein as such. If it is a smaller protein, you can directly use it. Or if it is a bigger protein, you need to chop it down into smaller peptide chains. Or this protein could be immobilized onto the blotting sheets. Mainly here, the blotting sheets that we use are made up of PVDF, that is a porous polyvinylidene fluoride membranes, and this immobilized proteins can be acted upon in the next steps using the Edman reagent. Okay, so this is the isolation or purification of protein using electrophoresis. So once that has been done, next step is denaturation. As I said earlier, you can either use high temperature denaturation coupled with acidic treatment, or treatment with thiol reagents or phenols to avoid complete degradation of the amino acids, and to ensure that the disulfide bonds are also broken to straighten up or to linearize the protein molecule. We know that during the secondary structure formation and all, there are disulfide bridges that are being formed, which helps the protein to fold. So now we need to break all those bonds so that the protein is now linearized, so that the N-terminal reading or the sequencing can be taken forward. Okay, so after the size separation or the separation in the gel electrophoresis, you can denature it also if it is a very big protein, and to straighten up the protein chain. So now is the actual process that is taking place, where we are using the N-terminal labeling. As I told earlier, here the reagent that we are using, it is PTC, the phenyl isothiocyanate. Okay, this can be added to either the peptide as such, or the adsorbed peptide that we have immobilized on PVDF membrane. And here, as I said earlier, this reaction is done under slight alkaline condition. So we will be using basic buffer solutions. And once this is done, the reaction takes place between the amine group of the N-terminal amino acid and the Edman reagent. So the terminal amino acid can be selectively detached by changing again the pH, where now we are shifting it to an acidic pH by making use of some kind of an anhydrous acid. And once this cleavage is done, the derivative now isomerizes to give a stable derivative of the amino acid, PTZ amino acid derivative. We call it as phenyl thiohydantoin. Okay, and once this is done, it can be fed to the chromatography system, and the whole cycle can be repeated with the shortened polypeptide chain till the last amino acid is read by the chromatography system. Okay, so that is the N terminal. So this is again what I have been speaking about. You have your polypeptide chain after degradation, denaturation, and this is the N terminal. Now you are reacting it with the Edman reagent, that is phenyl isothiocyanate. And once this is done, change in pH using anhydrous acid could give a cleavage at the first peptide bond here. So it leaves behind uh, combination of the Edman reagent with the first amino acid, leaving behind a shorter polypeptide chain. So this polypeptide chain can again react with the Edman reagent to complete the cycle of reaction. So the first one that has been converted will now get converted to a stable derivative of PTH amino acid derivative, which we call as phenyl thiohydantoin amino acid derivative, which is now fed to the chromatography to identify which amino acid it is. Okay, so this is the Edman sequencing method. So this is again the same thing that has been shown here, and you can see what kind of reactions are taking place and which all bonds are getting affected out of this. And this is your protein. This is now taken up as linearized into a polypeptide chain. High pH, it can be straightened up, and alkaline situation, it reacts with the Edman reagent. You have your phenyl thiocarbamyl derivative, that is the first derivative. Once this is done, you go for a low pH treatment where this cleaves the derivative at the first position, freeing the first amino acid, leaving behind the smaller chain. This smaller chain can again go back, react with the Edman reagent, and the reaction continues till the last amino acid is fed into the chromatography system. So purifying and recycling the remaining peptide fragment can be done through the Edman process. So these are all the details of it. You can go through this when you get time. Okay, so one amino acid at a time is removed from the N terminal of the protein by alternately reacting the protein with the Edman reagent and cleaving the resulting compound of the protein with the acid. So that's what I said, using anhydrous acids, you can cleave the compound from the rest of the polypeptide chain. Here you should know that in Sanger's reaction method, what happened was that this acid hydrogenation used to completely degrade the whole polypeptide. But in here, that is an Edman degradation sequence, only one amino acid is being cleaved from the whole polypeptide, leaving behind the intact peptide chain there to continue with the reaction. That means you do not need too much of the sample at every cycle or every step of the reaction to proceed further. So that is one advantage of Edman degradation method over the Sanger's method of protein sequencing. So after the isothiocyanate and amino acid complex has been detached from the polypeptide chain, identification of amino acids can be achieved by eluting them on HPLC and comparing it with a standard mixture to check the retention time in the HPLC. So this is HPLC system which we could use for performing this separation and identification of the amino acids that are part of the polypeptide that we are trying to sequence. So the amino acid derived from each cycle of the Edman reaction can be identified by comparing the retention time of the sample amino acid with a known standard amino acid, and thereby you can elucidate what are the amino acids that have gone into the formation of the particular protein we were trying to sequence. Then, to understand the structure further, we could also couple the whole system with mass spectrometry, wherein the mass to charge ratio of the particles will be analyzed using this mass spectrophotometry, and the separated amino acid derivatives could be analyzed by mass spectrometry. Okay, so this is how you will get a spectrum out of this once a whole process is completed. So first strategy for identifying an unknown compound is mainly by comparing the experimental mass spectrum that we got from the unknown sample against a library of mass spectra that we already have with standard compounds. So the standard solutions of amino acids are also used, and the resulting pattern could be compared with the newly generated data compared with the already known spectrum produced from the standard compounds. So that is how the data is being analyzed. And as I said earlier, Edman reagent and this Edman reaction is found to be highly suitable for polypeptide chains up to having up to 40 to 50 amino acids. So what happens if the protein that we are trying to sequence is beyond that, or which is having more than 50 amino acids? What could we do? As I said earlier, in that case, what we do is after purification of the purification of the protein, we go for fragmentation using specific enzymes. Trypsin, chymotrypsin are all certain enzymes that we use to chop down these lengthier polypeptide chains into smaller polypeptide chains. So here the beauty is that trypsin cuts between specific amino acids, chymotrypsin cuts at specific amino acids. So we know where all these go and cut, so that the fragments can be easily identified and they can be arranged together to see the overlapping sequences. So once the fragments are done, as you can see here, this is your cell or the tissue, this is your protein mixture. You can go for purification using gel electrophoresis, and if it is very big protein that we are going to work with, then you go digest into peptides using the enzymes I spoke about right now. And once this is done, you can separate out the mixture using liquid chromatography. We saw earlier HPLC could be one good option for doing this. And once it is done, you can couple it with the mass spectrometry, where the ionized peptide ions can be fed into the mass analyzer, where the mass charge ratio will be taken into consideration, and finally you will get your spectrum. And by comparing the spectrum that we have got with the library of spectrums of standard molecules, we can deduce the amino acids that were present in our protein. So this is how the whole thing takes place if we are chopping it down with different enzymes. For example, this one was chopped down by the trypsin, and this again by trypsin. This was done by chymotrypsin. So by putting them along wherever there is overlapping sequences, like here you can see here, this is an overlapping sequence. So you put it over here, then you have this sequence, again you put it all. So now you start reading from one end to the other, then you will get your final derived sequence. So sequences of smaller fragments may be overlapped and they can be analyzed for the complete sequence, and start reading it from one end to the other. Now you will have your complete protein sequence done. Okay, so that is how we make use of the Edman reagent to understand the protein sequence or the amino acid sequence present in a particular protein. So even if we say that this is a very good process or method to identify the amino acid sequence in the proteins. It do have its limitations. One thing is that we saw that it starts reading from the end terminus of the polypeptide chain. So what happens if the end terminal amino acid is already modified or is coupled with some other molecules? No longer can the Edman reagent go combine or react with that end terminal amino acid. So in such cases, this process may not work. So in such cases, we might have to take C-terminal reading for understanding the sequence of that particular protein. So it also requires the use of either a separate procedure to determine the position of the disulfide bridges as well. We may not be able to pinpoint where exactly the disulfide bridges are present. So that is again a problem with Edman sequencing method. Then we need pure samples of proteins. Maybe we don't need too much of proteins as we need for Sanger's reaction. Still, we need purity of samples. Then reading can be done only up to around like 40 to 50 amino acids can be read. So again, we will have to make use of some other methods to chop down the bigger protein molecules so that Edman reaction can be used for analyzing this. So that is these are some of the small limitations of Edman's degradation method of protein sequencing, even though it is still considered to be one of the most reliable sequencing techniques, having great advantage over the other sequencing methods. One is that the rest of the peptide chain after removal of the N-terminal amino acid is left intact. Other than the Sanger's method where the whole thing is degraded, here only the one amino acid is removed, cleaved at a time. So that means the rest of the polypeptide is still intact, which can further be taken up into the cycle to complete the sequencing in a very sequential manner. So that is the most advantageous feature of Edman sequencing reaction. So it is still considered to be one of the most reliable sequencing techniques. And I said nowadays we have automated protein sequences. So most of the automated protein sequencers which we have today, which help us read through the protein sequences within matters of time. So one of the disadvantages was that the Edman sequencing method was that the manual Edman sequencing method was it took a lot of time to go through all those processes and to read these amino acids. So the automation of this Edman reactions has taken away that limitation from the manual process, and this helps us to deduce or understand the sequence of amino acids much faster than the manual method. Here also, the protein sequencing is being done in three major steps. First is labeling, separation, and finally analysis of the amino acid. And all these things are done automatically, so that is the beauty of it. And this is a device developed by Edman and Geoffrey Beck, as I told earlier. And you can see here, this is for Edman explaining the process, the automation process to his colleague. And you can see the prototype of the sequenator, which he designed way back. And this instrument was designed to contain reservoirs to hold all the reagents required for the reaction cycles, together with the receivers for effluents and how to control the temperature, the pH, and all those things. And all these things were preset at time intervals so that one after the other, the reaction would continue taking place. That means it was all programmed. And The process embraced the coupling step with formation of phenyl thiocarbamyl derivation of the protein and the release of the N-terminal amino acid as anilino-thiazolinone. And these thiazolinones of N-terminal amino acids were automatically transferred to the tubes in fraction collector and then fed into the chromatography system or other suitable procedures, maybe coupled with mass spectrometry also. Finally, you will get your sequence ready for understanding. So that is all these things are being done automatically, and you get your results within a few minutes or time. So that's about how it is all being done. And speaking about the protein sequencing applications, as we said earlier at the beginning of the lecture, this can be used. Once you know the sequence, you could understand the structure and function of the protein. New protein biomarkers could be identified, as well as drug targets could be identified. Then we could construct the phylogenetic tree to understand the evolutionary relationship between different groups of proteins. Likewise, orthologous as well as the paralogous proteins, whether they are sharing a similar ancestry or whether they are coming from different ancestors, all these things could be identified so that functions of the genes or proteins of unknown proteins could also be deduced from what we already know. So they can also be used to make probes for sequencing the cDNA library, as well as they could be used for protein engineering studies as well. And so these are some of the applications of protein sequencing, and they do have clinical applications as well. As I said earlier, we could go for newer antibiotics. We could find targets for drug molecules, then evolution of history of that particular protein. So we know that nowadays we are going through the pandemic when we hear that the coronavirus has been like coming up with different strains. So what is happening? What is the code protein like? Are they all similar? Do they have a similar structure? Do they have a similar sequence? So all these things also we could understand if we can go for protein sequencing, and the prediction of the cellular location of the protein based on its target sequence will also help us understand where these proteins are. Okay, the prediction of the sequence of the gene encoding that particular protein will also be possible once we understand a similar protein. Okay, so that is how when the microorganisms are mutating, we could say that, okay, if this is a case, this could be how the protein will be looking like once it is getting translated, the gene is getting translated. So all these kinds of predictions is also possible, and discovering the structure and function of protein through various computational methods. We know bioinformatics is playing a key role in deducing so much of information about genomics and the proteomics. So all these places, we can make use of the protein sequences of the known protein as well as the unknown protein, and comparing it with each other, we can predict the function, the structure, etc., of those proteins. So in a nutshell, we have been discussing about what proteins are, how the protein sequencing is to be done, or how can we do the protein sequencing. Especially, we have been speaking about the N-terminal labeling, where we are using Edman reagent to understand one at a time the amino acids of the polypeptide chain using the Edman reagent. So this process is known as Edman degradation method of protein sequencing, and we also discussed about the various applications of protein sequencing. So thank you for listening and have a nice day.